Okay, we'd like to get this next session started. Please come and sit down as quickly as you can. Okay, we'd like to start the next session. Please, if you could take a seat, we'd appreciate it. We're running a little over, so we just want to keep on the schedule. This is session 3, Network Effects of Synaptic Plasticity. We'll have 4 speakers, so I think it's going to be a great session. Our first speaker of the session will be Indira Raman, who is a professor in the Department of Neurobiology at Northwestern University, and will be speaking about synchrony and synaptic signaling in cerebellar circuits. Thank you.
Thank you. Thank you for inviting me, especially Mark and Kim for arranging this and for inviting me, and for getting through my tongue twister of a title here. So today I'd like to tell you about synchrony and synaptic signaling in cerebellar circuits. And in particular, I'd like to talk about how my lab has been using the cerebellum to study neuronal coding. I guess I can point like this. And so here we see a parasagittal slice of the well, you don't see my pointer, but of the mouse cerebellum. And to the left is what my students like to call my egg diagram of the key cells in the cerebellar circuit. And today I'll have most to say about the Purkinje cells, they're in pink, of the cerebellar cortex and their targets in the cerebellar nuclei, particularly the large cells that project out to premotor areas and which form the output of the cerebellum and whose activity can lead to movement. But since this is actually my first Cosyne meeting, I thought it would be kind of fun to go back to an early computational neuroscientist, in particular David Marr, and to quote a paragraph from his 1969 paper, Theory of Cerebellar Cortex, which really provides the best introduction to the talk that I'd like to give you today. And so he starts out saying, in this paragraph, he starts out saying that the main branch of a Purkinje cell axon goes to one of the cerebellar nuclei and forms the only output from the cortex, and he draws attention to the fact that its effect in the nucleus is inhibitory. So he's already noting that this is weird to have an inhibitory projection neuron. And he says it's an assumption of his theory that the CNS has a means to convert a signal in a Purkinje cell, even though it's inhibitory, into an order to provoke an elemental movement. And he says that inhibitory nature of the Purkinje output suggests that it may require a positive effort to read from the cerebellum. Something special has to go on for this to matter, since excitation must be fed into the circuit somewhere. And he suggests that maybe this is coming through some form of excitation. And so there in the lower left, you can see MF, the mossy fibers, which bring integrated sensory input and motor command-related information to the cerebellum. They excite the cerebellar nuclear cells, and they disynaptically excite the Purkinje cells. And so he suggests that this would be useful if it were required that cerebellar output should often be ignored. And the fact that cortical output is inhibitory can be interpreted as a convenience for easy ignoring, right? And this is, of course, the source of all the cerebellar jokes, that you don't really need a cerebellum, which is not wholly true. But there are some weird things about the cerebellum, because when we look at the circuit, the Purkinje cells should exert a powerful inhibitory control over their targets in the nuclei. And yet in classic work like this and other papers from Armstrong and Edgley, 1984, you see that when measurements are made of Purkinje cell firing rates and cerebellar nuclear cell firing rates, in this case when animals were walking on a treadmill, cats were walking on a treadmill, we don't see the inverse coding we would expect from a powerful inhibitory control. Instead, we see a co-modulation. And so that raises a puzzle, and this is the question that's been driving my lab for a long time. How is it that Purkinje cells actually do something, that they make cerebellar output reactive or predictive or generative, and they do it on a millisecond time scale, right? That's the other hallmark of cerebellar function, that it's very fast. And I'm actually trained as a ion channel biophysicist, and so we've been starting really from a reductionist point of view from the ground up, starting out looking at intrinsic properties of how cerebellar neurons fire, and then building up to understand some synaptic properties about the transformations at cerebellar synapses, and then finally bringing it together into circuit properties to try to understand how cerebellar signaling in vivo emerges from cellular specialization. So I'll start with just a little bit of biophysics, showing you a cerebellar nuclear cell, which has a specialization, like the afferent Purkinje cells, of firing action potential spontaneously. These cells will fire 50 to 100 spikes per second. This is fast, with no synaptic input. So we want to know how, and it turns out that these cells have the simplest possible mechanism for spontaneous firing, and that is that they simply rest above threshold, right? And that's a specialization. They have a tonic cation current that depolarizes them. So if you put TTX on a firing cell, as is shown there, you see that that particular cell rests at minus 50 millivolts above threshold. And on average, you can see below, the resting potential is about minus 40 millivolts. So if you think about what happens when the cell fires an action potential, when it's done, it will seek its resting potential, but in doing so, it'll pass through threshold and will make another spike. And so these cells will always be making another spike. They can't be hyperpolarized below threshold by their own intrinsic currents. They can only be hyperpolarized below threshold by extrinsic current that comes in the form of synaptic inhibition, and that comes from Purkinje cells. All right, so we can look at the characteristics of the Purkinje cell-mediated inhibition. Up there in the left, you can see some iPSCs that were recorded at different temperatures in a cerebellar nuclear cell in a slice from stimulating the Purkinje cell afferents, all right? And so I should mention that there's a big convergence of about 40 Purkinje cells on each cerebellar nuclear cell. So when we evoke those spikes, we're actually using an electrode to stimulate them all simultaneously or synchronously, right? And we see that they're actually pretty big. They're big iPSCs. And so with 2,000 or more iPSPs per second, you know, it's kind of amazing that these cells can fire, the nuclear cells can fire at all. But the other thing we notice about these iPSCs is that they have a specialization of being really fast. All right, the time constant here for these GABA-A receptor-mediated synaptic currents is 2.5 milliseconds, and that's about 10 times faster than you see in many other places in the brain. So when we see that specialization, we think maybe something interesting is going on. So even though these are very big currents, they're very fast. And so if you use a stimulating electrode to stimulate the Purkinje cells synchronously at about 50 hertz, you see that profile of a series of iPSCs, which then depress by about 50%. But the interesting thing I want you to notice is that the current decays fully in between stimuli, right? Not surprising, 8 time constants. But it brings us back to that idea that I just mentioned, that the nuclear cells can't be silenced by their own intrinsic current. They need extrinsic synaptic current. So it raises the possibility that if you activate a bunch of Purkinje cells simultaneously, you'll get a big synaptic current, but then it will decay completely, and there might be a little window right afterward when the cerebellar nuclear cells can fire. And we can test that in the slice and measure with all excitation blocked, right? We can stimulate Purkinje cells at 60 or 70 or 80 spikes per second. Those are the little carrots. And you can see the action potentials that are entraining to the stimuli, even though this is an inhibitory input, right? And so if we look at the plot of the median firing rate against the iPSP rate, right, the output against the input, you see that when we stimulate in red synchronously the Purkinje cell afferents, we always get a high firing rate, sometimes perfect entrainment, sometimes doublets at the low frequencies, sometimes we skip a cycle at the higher frequencies. But we always get a big output that is much greater than what we get when we use desynchronized inhibitory input, which we can apply through dynamic clamp. Just 8 afferents are sufficient to silence the nuclear cells no matter what rate you stimulate at. Okay, so this teaches us that cerebellar nuclear cells actually have the biophysical capacity to detect and respond to Purkinje cell synchrony. All right, but this is biophysics, right? We want to get a little more physiological. We can think about the excitation coming in. And so Yichun Wu, a grad student in the lab some years ago, tried to get to more physiological conditions, but still in vitro, by measuring the properties of all the excitatory inputs from the mossy fibers, and he could mimic the magnitude, kinetics, short-term plasticity, convergence, basal firing rates of the mossy fiber EPSCs and the Purkinje iPSCs, and then combine them in dynamic clamp. And when he did that, you got currents like action potentials like we can see above the plot. In black, we see what happens when he combined asynchronous excitation and inhibition. And then in green, you can see what happens when he has the same amount of excitation and inhibition per second, but he synchronizes half of the inhibitory inputs, and what you see is more spikes, right? And that's plotted down below for a range of EPSP rates, right? It's an input-output curve of spikes against EPSP rate. The black is with all the excitation and inhibition asynchronous, and the green is what happens with half the inhibition synchronized. You always get more bang for your buck, right? And this is not a discovery, right? This is a demonstration. It has to be this way because the Purkinje cells fire, you get a synaptic current, the current turns off, and then you have a little window where there's less inhibition. So the idea is simply that the efficacy of excitation can be controlled by the spike timing of inhibitory inputs, right? And so it raises a possibility that if Purkinje cells were to increase their synchrony in vivo, right, then the mossy fibers might more effectively drive nuclear cells, right? And the converse would also be true. If Purkinje cells were to decrease the synchrony, then we might more effectively cancel cerebellar output. Okay, and the reason we care about this at all, or it's worth considering, is that ever since Marr's paper in 1969, the line of experimental research has been showing us repeatedly that simple spike synchrony can occur in vivo, right? It's been seen many times, but it's always often been incidental because it's very hard to trigger any kind of synchrony. So what we really are interested in is synchrony a variable in vivo? And so to explore this question, my then grad student, Spencer Brown, now postdoc in the lab, started recording from Cruce 1 and Cruce 2 of the cerebellum, regions of the cerebellum that are associated with whisker movements. A big chunk of the mouse cerebellum is dedicated to whisker-related behavior. And he could make loose cell-attached recordings from the wake head-fixed mice, and he could then trigger a reflexive protraction of the whisker by a 10 millisecond puff of air to the whisker pad. And so you can see examples of those cell-attached recordings. There's a complex spike in the middle. That's how we identify these cells to be Purkinje cells. And then comes the position trace, where you can see that there's a puff of air applied to the whisker pad. It briefly deflects the whisker backward, and then upward is the protraction. Right? And below, you can see the action potentials that are generated in a single Purkinje cell, and then below that, we convert that into an instantaneous rate trace. And I want to draw your attention to that time scale there, right? We're looking at a few hundred milliseconds across that whole time scale. Okay, so he can do that in many cells. Here are examples of these instantaneous rate traces in many Purkinje cells. We can average them, and we get something that's very similar to what many other people have seen, right? In red, you can see the population rate trace of the Purkinje cells, and we're interested in the population, again, because of this convergence that we know occurs. And you see that it matches quite nicely the whisker protraction trace in black there, right? There's an offset of 15 milliseconds, but if you account for that and plot the one against the other, you see this very nice correlation. And, this is very similar to what many people have seen with many kinds of movements, reaching, locomotion, and so forth, that there's a match between the simple cell's firing rate and the movement kinematics, and this is why we agree with many people in the field who we all agree that there's a movement-related signal in the simple spike rate. Okay. But what we were really curious about in this experiment was, does cerebellar output really matter for something like this reflexive whisking? And so these Purkinje cells were expressing channel rhodopsin, and so Spencer could indiscriminately illuminate these regions of the cerebellum, which would activate a whole bunch of Purkinje cell firing at random. And that was effective at shutting down this region of the cerebellar nuclei, and we wanted to see what happens to the whisk. And that's shown here. In black is the position trace, again, of the whisker movement under control conditions evoked by the puff. And in blue, you see what happens when we shut down the cerebellar output. You get a whisker movement that's about half the size, or reduced by 40% on average. Right? So it's always a pleasure to find out that the cerebellum really does matter for something. But there's a puzzle that's presented here, because it suggests that the output of the cerebellum, those cerebellar nuclear cells, are increasing their firing under normal conditions, so that when they decrease their firing, we see that the whisker movement is smaller. But we've also seen that the Purkinje cells have increased their firing rate, and so we're kind of back to the 1980s with the same old paradox of the idea that cerebellar nuclear cells and Purkinje cells are increasing their firing together. So we made recordings from the nuclear cells, and in blue you can see the instantaneous rate trace from a bunch of nuclear cells, and you can see indeed it does go up with the puff. But now I want to draw your attention again to the time base and say that it's expanded, and the red trace and the black trace are the same as the ones that are shown up higher, right? Where I showed you the great correlation, but you can see early in those first 50 milliseconds there's some extra stuff going on. right? And it's hard to interpret with that rate trace, so we thought, well, let's just look at the timing of these spikes. And we replotted the same data. This is many trials in many cells, nothing's selected here, plotted as a PSTH, right, a peristimulus time histogram. And you see that in that first 50 milliseconds, it's true that the number of spikes in the Purkinje cells and the number of spikes in the cerebellar nuclear cells really do go up together, all right? But this is a moment when we can actually go back and think about what we learned in vitro, right? What we know about the synaptic transformations here. We've learned that things are happening on a single-digit millisecond time scale, not tens of milliseconds. And so maybe this isn't really the best way to look at these data. And so we wanted to go back and take the same data, and instead of looking clumped together in 50 millisecond bins, we wanted to look in 2 millisecond bins, right, where 2 milliseconds is smaller than an inner spike interval. So what we're really looking at is spike timing, okay? So here are the same data from the Purkinje cells, except for we've split the complex spikes there in that greenish yellow, riding atop of those gray simple spikes, okay? And what we see, and the data are plotted as spikes per trial per bin, so you can multiply by 500 to get the spike rate. The cells are firing along at about 75 spikes per second. But what we see then is that the first thing that happens after that puff is applied at time 0 is that there's a concerted decrease in the probability of firing in the Purkinje cells, all right? It goes down by about 30% for just 4 milliseconds. It's a very well-timed event. And then we get an increase in spike probability, right? And then a decrease, and then we have a bunch of complex spikes, and then we get a slow, non-well-timed increase in the firing of the simple spikes, and that's that rate change that follows the whisker protraction. Okay? And so then when we see what is happening in the cerebellar nuclear cells, we get I should mention that this is the signature of synchrony, right? Because the cells are doing the same thing at the same time. And I'm using a very simple definition of synchrony here, okay? The thing that's a surprise is that it's starting with a synchronous suppression of simple spikes rather than an elevation. But it doesn't actually matter because the signal seems to be there. So if we look at the cerebellar nuclear cells, we can see that those cells are showing a very concerted increase, a drop, and another increase in firing. And if we overlay those two, you can see that the cerebellar nuclear cell increase does correlate in time with when that disinhibition is happening, that is the drop in the firing of the Purkinje cells. That's not the only thing driving it, though. Since we're making loose cell-attached recordings in vivo, we can also get a measurement of the mossy fiber excitation that the nuclear cells are experiencing, and that's shown there as that cyan wiggle. And that first transient is the response to the sensory input, right? And the later wiggle is happening at the time of the whisking-related motor command. And so we think that that's the motor signal. And so the thing that I find interesting about this, or that was revelatory when I looked at this, was that every time I've looked at my egg diagram with the excitation from mossy fibers and the inhibition from Purkinje cells converging on the nuclei, I've always thought of those as necessarily being in opposition, right? The balance of excitation and inhibition, or the imbalance, as the case may be. But here we're seeing that under these circumstances, at least, the excitation is working with disinhibition to give us a very large output. Okay? And so it looks like the excitation is starting to drive that cerebellar nuclear cell spike probability high, but it's going to be aided by these well-timed drops in inhibition. Okay, that's with a puff of air to the whisker pad. What if we use a slightly different kind of whisker stimulation? Here, what we did was, you're looking down in the photos below, pictures below. We're looking down on the head of the mouse, and you can see the two whiskers that are remaining after trimming. And we're also looking down on a bar, a vertical bar, that's on a motorized manipulator that can move silently in the dark. And so we can make that manipulator move forward, make contact with the whisker, and then deflect it backward. All right, that's what we call a passive contact. All right, and way up high, you can see that passive contact showing us the whisker position. You see it goes downward because the whisker is deflected backward, all right? We can also just have that bar present, and the mouse can whisk against it in the dark. That's below, that's the active contact. And so the nice thing about this is that it's really a whisker-specific stimulus, and we don't have this reflexive protraction afterward. And so in the PSTH, you can see in blue the response of the Purkinje cells, right? Again, many Purkinje cells, many trials per Purkinje cell. This is the cumulative, the population PSTH. And you see again that the first thing that happens, really 8 milliseconds after the time of the contact, is a 4 millisecond suppression in Purkinje cell spiking. And then the firing rate goes back up, firing probability goes up a bit, but we never get that big increase, presumably because we're not getting that forward whisker protraction. And then superimposed, obviously, is the cerebellar nuclear cell response. And again, you see that there is an increase, that correlates in time with when we have disinhibition. So the satisfying thing here is that we're finally able to see the powerful inhibitory control that ought to be there, but is very hard to extract in many kinds of experiments. All right, so here when we're syncing to the sensory input, we can see that. Right? If we look at the active contact, you see that the Purkinje cell basal firing rate, it's plotted here as 2 millisecond rate, right? I multiplied by 500 here, is higher to begin with, but then again, just 6 to 8 milliseconds after the contact, we see this 4 millisecond suppression in Purkinje cell spiking, and it correlates in time with an increase in the cerebellar nuclear cell firing probability. And we can, illustrate that inverse relationship by plotting the bin values of the cerebellar nuclei against the bin values, the corresponding bin values of the Purkinje cell. And in the open symbols, you can see what it is for the passive contact. It's a very strong correlation. But of course, that correlation is driven really by the 2 bins that represent that transient response. Likewise, in the solid symbols, you can see the relationship for the active contacts. Again, there's a correlation that is driven by the couple of bins associated with that transient response. The thing that's interesting about that graph, though, is that the plots are offset on the axis. There's not some fixed relationship between cerebellar nuclear cell firing rate and the Purkinje cell firing rate, right? And so you can see that even more dramatically here. If I show you the same data, but instead of just showing you 80 milliseconds, plus minus 80 milliseconds around the time of the stimulus, as I'm doing at left, here at right, I'm showing you plus minus 500 milliseconds associated with the stimulus. And let me reemphasize, these are all the same Purkinje cells and all the same nuclear cells in all these experiments. Right? And so what we see in the active contact in the upper right is that as the whisker protracts forward to make contact with the bar, the Purkinje and cerebellar nuclear cell activity is actually co-modulated. They're going up together. And it's only the vicinity of the contact that we start seeing a divergence. All right? And likewise below, when we look at the passive contact, if you look carefully, after the passive contact, the Purkinje cell firing rate is actually higher than it was beforehand, but the nuclear cell activity is not reflecting that in any way. Okay? And so what we're seeing is that the nuclear cell firing rates are actually varying inversely only with the transient, but not the prolonged Purkinje responses, okay? And so this is like an AC-coupled synapse, right? The high-frequency signals, at least under these stimulus conditions, are being transmitted, but the low-frequency signals are being filtered out, right? And so those high-frequency signals are actually coming from the synchronous sensory input, right? Their sensory input is evoking synchronous signals that are greatly changing the nuclear cell output. But these asynchronous motor-related signals are having less of a dramatic effect, right? So what we really want to know is how. How can this simple spike suppression arise? Because those of you who are familiar with the circuit or who noted it at the beginning will recognize that you don't really predict from the anatomy of the circuit that the first thing Purkinje cells are going to do with a sensory input is to suppress their firing. All right, so here's a slightly expanded version of the circuit. Down in the lower left is the trigeminal ganglion cells. These are the cells that are transmitting the sensory input from the whisker. They excite the trigeminal nuclei, right? And the axons of the trigeminal nuclei are actually mossy fibers, which go to the cerebellum and excite the nuclei and excite the Purkinje cells disynaptically, and there are a couple of feed-forward inhibitory loops there involving Golgi cells and molecular layer interneurons in the way upper left. Okay, and so if you just look at that, you expect that the excitation should reach the Purkinje cells before the inhibition. So how does this work out exactly? So Meghna Halla, a graduate student who just finished her PhD in the lab, decided to go back to synaptic physiology in the slice. And she made recordings from Purkinje cells, whole cell recordings from Purkinje cells, and then she could stimulate parallel fibers so she could the axons of granule cells, right? So she could monosynaptically evoke EPSCs and disynaptically via the MLIs evoke iPSCs, right? And so what we're looking at here are EPSCs in red, which she could measure at minus 70 millivolts. And what you see is a reflection of what other people have seen before, namely Wade Regier, that these are among the most strongly facilitating synapses in the brain, right? The EPSCs start small and then they grow. That's a stimulus at 50 hertz. But if she flips up to 0 millivolts, so she can record the iPSCs evoked by the same inputs, right, the same parallel fibers being activated. She sees iPSCs that are rock solid, right? And this was true cell to cell, right? Those are individual cells. Below is the average of some 50 cells, but this was consistent on a cell-by-cell basis. And so if you think about that, it means that that EI ratio is necessarily going to be changing, right, with repeated stimulation. And at the onset of stimulation, we're expecting that the excitation will be smallest with respect to the inhibition. And so that raises the, and this is an interesting specialization also, and so it suggests that disynaptic inhibition may actually outweigh excitation, even though the excitation gets there first, but the effect may be to suppress Purkinje cell firing. And so Meg and I can look in vivo in mice that are expressing channel rhodopsin in granule cells, and again, she can record from Purkinje cells and indiscriminately activate granule cells many times and make a PSTH. And this is the population PSTH, but split into two categories. In light blue, you see that the majority of cells, 60% of the cells, actually respond to granule cell stimulation in vivo with a suppression. That's the thing that happens first, right? And in dark blue, you can see that some of those cells, a different set of cells, about 40% of the cells, do respond with an excitation first. So we can get both kinds of responses, right? But it isn't crazy to say that inhibition can happen first. And in fact, most recently, Theo Rossi, a postdoc in the lab, has expressed channel rhodopsin specifically in the trigeminal nuclei. And so he can now record from Purkinje cells and stimulate the mossy fiber afferents that come from the trigeminal nuclei. On top is the principal nucleus, and in the middle is interpolaris. Both of these are carrying whisker-related sensory information to the cerebellum. And those are population PSTHs of 60 on top and 50 Purkinje cells. And you can see the net effect is suppression with stimulation of these sensory afferents, right? And that's very different from what you get in the bottom graph, where he has expressed channel rhodopsin in the pontine nuclei, which bring motor command-related input and sensory input to the cerebellum. And there, when he stimulates, the population response gives you the excitation that is, again, what you predict from the circuit. And only in about 12% of the cells does he actually see an inhibition-only response. So again, this seems like something that is really typical, at least, of these sensory afferents. Okay. So the idea, then, is that synchronous inhibition of Purkinje cells may actually arise from especially the unfacilitated granule cells that are activating intervening molecular layer interneurons, and it makes us become interested in how those molecular layer interneurons are responding to the stimuli. But I should also admit right now that I've been talking about synchrony, or trying to get you to consider the possibility that synchrony is an important component of signaling, but everything I've told you about has been recording from one cell at a time, right, and then triggering to some stimulus, right? Ideally, we wouldn't record individual neurons in multiple trials, but we would record many neurons on each trial. But that turns out to be difficult to do. Multi-electrode arrays have promise, but they have some difficulties associated with identifying cells and getting as many cells as we would actually like. There's calcium imaging, of course, which lets us have access to many, many cells, but most of the calcium indicators are too slow at present for the rates of firing that we're seeing in the cerebellum and that we're interested in. Genetically encoded voltage indicators are fast enough and hold promise, but the challenge there is the signal-to-noise ratio. And so recently, Spencer Brown, who's returned to my lab as a postdoc, has been developing methods to overcome this, to deal with the signal-to-noise ratio. And he's developed methods for high-resolution one-photon voltage imaging at 2,000 to 4,000 frames per second in awake head-fixed mice. And so he's using the indicator Force 1F from François Saint-Pierre's lab, and up there in the left, you can see an individual molecular interneuron. It's a DMD-targeted illumination, so you can see that particular cell. And then you can see some examples of the action potentials that he's recording in vivo in the awake mouse, sampling at 3 kilohertz. And far to the right, you can see the overlay of all of those many of those action potentials. And you can see that the spike width is about 1 millisecond. So we can really see what we were interested in seeing. And the nice thing about this approach is that he can record from a whole array, here a couple of dozen, of molecular layer interneurons that are sparsely distributed in the cerebellum. And on the right, you can see the corresponding action potentials occurring in each one of those 26 molecular layer interneurons. And so this was one of his early attempts. Now he's improved his technique. In this particular case, he's recording with PACE from Madhukanan's lab. And this is his pièce de résistance, keeping with the theme of everyone using a little bit of French in their talks. And so here he's got 318 neurons that he's recording at once. And there he's applied a puff, and I think you can see that there's something happens in these neurons with the puff. And so there's the spike raster of the 318 neurons, and then he can just measure, right, see how many cells are coactive at once. So we kind of need to define what window we were interested in for synchrony, and so what we did here was Spencer plotted just this peak coactivity, you know, what fraction of the cells were firing at the same time, as a function of bin width. Starting at 1 millisecond, you can see that it increases linearly up to about 4 milliseconds, then it becomes sublinear. So we're taking 4 milliseconds as our window of synchrony, and that actually matches nicely with what we've seen, of course, in the Purkinje cells. Okay, so now he can look at what is the average synchrony or coactivity on each trial, averaging here across trials. On average, it's about 40% synchrony, and these fields are about 100 on average, 100 cells on average. But what's kind of interesting is that we see that it varies from trial to trial, right? We've never been able to look at trial-to-trial variability, and here we can see that it can be as low as 10 to 20% synchrony of molecular layer interneurons, and as high as 80 to 90% synchrony. And we thought, well, that's nice. You know, maybe we can actually use that as a tool to understand or figure out a little more about the relationship between this synchrony and the outputs. Okay, so what he did then was do many trials, right, but then calculate the coactivity on each trial. You can see it's varying by the varying brightnesses of that yellow there, right? And then he can look at the associated whisker movements. And that's from one field of view. He can do this in many fields of view. And then he can bin the amount of synchrony, right, so that he can take the trials that have 10% synchrony in the darkest colors, coldest colors, and then up to 80% or more synchrony in the warmest colors. You see the rainbow on the above, which is showing us increasing mean coactivity. And then he can collect the whisker movements associated, right? And then those are shown below, and you see again the rainbow pattern, right? So there's some relationship there. He can plot the cumulative amplitude just to have some metric to relate to the amount of coactivity, and plotting that whisker movement cumulative amplitude against the maximum coactivity gives you a very high correlation. So we have a very nice correlation here between the amount of molecular layer interneuron synchrony and the whisker movement, but of course we're curious whether we can get any evidence for causality. So the experiment that Spencer did then was to record whisker movements in mice expressing channel rhodopsin, in this case in Purkinje cells, and then make use of what we've learned about the very stereotyped windows when we see the synchronous suppression of Purkinje cells, right? So we know that there's this 4 millisecond suppression of Purkinje cells. We know when it occurs, right? And so what he can do is apply channel rhodopsin for 20 milliseconds as opposed to the very long stimuli we gave before, so that he can override that suppression of simple spikes in the Purkinje cells. And what you can see in the upper set of whisker traces, the black is showing you the whisker protraction under normal conditions, and the blue is showing you what happens when he overrides the simple spike suppression for just 20 milliseconds. And what you see is instead of seeing that upstroke, that's the forward protraction, there's almost nothing. And then later we see a protraction, but that's actually an offset response to when we turn off the channel rhodopsin light, the illumination, right? Anytime, if we extend that illumination, we always see that response afterward. So the real observation is that you don't get that upstroke if you prevent that suppression in the Purkinje cells, right? Now, if you just move over the time of stimulation by 20 milliseconds so that you get the suppression and then you activate the Purkinje cells, that's shown below, and there's really no change in that whisker protraction. Okay, now he does, so that it seems like that suppression is necessary. Then we turn the corner and we use mice where the channel rhodopsin is in the molecular layer interneurons. And now he's taking advantage of the fact that he knows exactly when the molecular layer interneurons will synchronize, and he knows that on average we have about 40% synchrony. So now he gives a 5 millisecond illumination to boost up that amount of synchrony. So in black, you can see the forward protraction of the whisker, and then in blue, you can see that that's enlarged when he illuminates the molecular layer inner neurons just for 5 milliseconds to increase their synchrony. That's when we have a puff, right? Below, what you can see is what happens when there's no other stimulus, no sensory stimulus, but he just gives a 5 millisecond stimulus to the molecular layer inner neurons, and you can see that that is sufficient to elicit a small whisker protraction by itself, right? Okay. So overall, what we're imagining here is that in red, we can see the PSTH of the molecular layer inner neurons across all trials. And so what we're thinking is that the sensory input is organizing the molecular layer inner neuron activity, so that goes up, right? And then those MLIs can converge onto Purkinje cells, and that will cause that synchronous suppression of the Purkinje cells, which then will work with the MOSFET fibers to drive the nuclear cell output high. And again, a key reason why this kind of a code may work here is because each cell is sampling the population of its afferents, right? And so we can think about, rate codes, in which, of course, a single cell can make more or fewer spikes over time. But to get a rate code, it requires integration over time, right? So you can have slower responses. Even if they're fast and absolute scale, they're slower because you'll need at least a couple of spikes to make a rate code. Here, we have the same sort of thing, but over a population. The synchrony code, in a synchrony code, the population of cells can make more or fewer spikes in a short time, and that is well suited for giving these very rapid millisecond scale responses. So if we go back to the points that Marr brought up in his paper more than 50 years ago, where he says the CNS may have a means to convert an inhibitory Purkinje cell signal into an order for a movement, we can bring up some of these specializations that we find, like the short-term plasticity of the parallel fibers and the MLIs, which are rock solid, right, which favor Purkinje cell simple spike suppression exactly at sensory onset, right, and then those big but fast iPSCs in cerebellar nuclear neurons, which can fire during synchronous gaps in inhibition. Right? He says that it may require a positive effort to read from the cerebellum, and so it looks like, with tactile stimuli at least, Purkinje disinhibition works with excitation to drive nuclear cell output and augment the whisks. And this would be useful if cerebellar output should often be ignored. Well, we see that during whisking movements, and particularly during spontaneous movements, which I didn't show you, the non-synchronous excitation and inhibition can cancel each other. And that's what makes these cerebellar circuits act, at least under these conditions, as high-pass filters. Right? So the working hypothesis we have is that in complex movements, maybe deviations from intended execution may evoke unexpected tactile sensations, and then sensory input may recruit particularly inactive granule cells, which could favor Purkinje synchrony that might help produce corrective movements. So I'd like to acknowledge the people in the lab who contributed to the work. I wish I could point to them, but I can't. But so Yichun Wu, second row, second in. Fourth one in, Spencer Brown. Sixth one in, Mauricio Medina. Seventh one in, Megan Ahala. And third row, third one in, Abby Pearson. And last row, first one in, Dan Paget. So thank you for your attention. But before I end, I will make a plug to come continue this conversation in, not this particular one, all conversations, in Chicago at the National Institute for Theory and Mathematics in Biology. There's a booth, and please come get more information about that. So thank you for your attention.
Fantastic talk. We have about 7 minutes or so for questions. So if you have a question, please come up to a microphone. Yes?
Thank you, Indira. I was wondering, you referred to this dichotomy about the synchrony code and the rate code towards the end. And I was just wondering, recently, given that in behavioral timescales, there's been this these reports of, uncanny heterogeneity, mixed selectivity, multiplexing, almost rivaling what we observe in the cerebral cortex. Is there, how do we take these, here you mentioned that these non-synchronous patterns could cancel each other out, but surely there must be something coherent going on at the population level that drives a downstream response.
Yes, and so how can you have multiplexing under these circumstances?
Well, I was wondering if you might allow for some additional plasticity at the nuclear synapse.
Oh, absolutely. I mean, I think that what we're seeing here is some bare bones descriptions, right? I left out a number of elements, partly because it's a 35-minute talk, and I've left out a number of cell types, and so anyone with real familiarity with the cerebellum will be thinking about what all these additional cell types are, in addition to the heterogeneity that you have even at these common cell types. The thing that I find interesting here, the reason we're following this, I mean, if I could get away from Synchrony, I would do it, believe me, but it just keeps showing up. And the more we want to get away from it. And the thing that's interesting is that it seems like a very robust response that is organized under a very specific condition, right? And what I'm curious about now is how broad will the range of conditions be that organize the Purkinje cell activity in this way? Because I still, again, agree with, or am inclined to assume the idea of many people in the field that we would have this rate coding, the population rate coding, where everything is coming together and giving us some kind of more smooth modulation, and that may happen under learned behaviors. I think what we're looking at here may be something that is a trigger for learning, or that happens when the unexpected thing occurs, which is very often a thing that drives learning. We're looking at a very boring case of the reflex, very simple behavior because it helps us separate sensory and motor, which was what we wanted to do, again, coming from the ground up. But I suspect that might be something that would be, in a more learning suitable condition, something that would drive changes so we get more smoothly modulated responses. Does that make sense?
Thank you.
Question back there?
Thanks, Indira. I thought that was a really beautiful description of how having really detailed knowledge about the circuit can help explain some very unexpected phenomena, so that was great. Since you mentioned other cell types and you also mentioned learning, I'm wondering what your current thoughts are about climbing fiber inputs to cerebellar nucleus neurons and what role those might play in the kinds of synchrony and other features you were talking about today.
Yeah, climbing fiber input particularly to the nuclei?
Yes.
Yeah, so that's a great question. So we've measured characteristics of climbing fiber inputs to the nuclei. All I can tell you is that they're there. They are not activated, as you know, at as high rates. What I'm very interested in is the fact that boy, this is hard to look up there. I'm going to look down, even though I'm still talking to you. So the answer is I don't really have much to say about them, but what I find striking is that the climbing fiber input is later, right? It's a lot later than the suppression that I'm talking about. And so it seems to me that the suppression that I've been observing in the Purkinje cells is what you do on this trial, and that the climbing fibers that are input that's happening later on, maybe this is how we're going to get ready for next time if it's more of a learning-related situation. Now, whether that excitation coming in the to the cerebellar nuclei is also helping contribute to balancing the inhibitory input from the Purkinje cells, I don't know, but it seems like a plausible idea.
Thank you.
We have time for one more question. Yep.
Thanks, Indira. Question. So in the first part of your talk, you showed that before the active movement, we have gradual increase of Purkinje cells.
Yeah.
But then you also showed we have gradual increase of MLI before.
Yes.
So how do we?
Yeah.
Yeah.
Right. This is this AC coupled thing. I don't understand that. I glossed over that. But if you look carefully throughout all of these things, the only time you see the expected inverse coding where inhibitory neurons inhibit their target cells is in these transients. At the other points, during the forward protraction, in the active contact and in the forward protraction that's sensory evoked, you see the MLI activity go up in parallel with I can't use the word parallel, along with the Purkinje cell activity, and you see Purkinje cell activity go up along with the cerebellar nuclear cell activity. And that's why I'm saying that there is something going on that is making these rate changes that are more protracted in time not get transmitted in the expected inverse coding kind of way. It's just the observation, right? How? I don't know. I'm imagining that there is a net excitation and inhibition from all of these cells firing simultaneously in the various loops that may be just biased toward excitation at all these levels. But it's exactly what you need for cancellation or a little bit of output, right? And that's why I think that it fits together like that.
Could it be that MLI inhibition is less synchronous at the time of contact? But you have access to that data.
We can see it. It is synchronous at the time of contact.
But less so than before the contact?
Before the contact. I'm sorry. So the molecular layer, the rate is elevated after the contact, and so then you end up with sort of the same old puzzles of the rate and the synchrony, right? So the firing rate before the contact is at one level. Then we have the contact, the molecular layer interneuron response goes way up, and then we have this increase, but that has no temporal pattern to it that we can find, right? But that isn't reflected in the Purkinje cells, which also show that, which suggests that they're getting excitation, presumably from parallel fibers, that is also driving that. Right? So it is a puzzle, but that's why the thing that comes through reliably is the AC coupling, is the high-pass filtering. The rest of it, still a mystery. That's when I should get off the stage.
Great. Yeah, let's thank Indira again. Our second speaker is Samuel Eckmann. He's a Royal Society Newton International Fellow at the University of Cambridge, and he'll be speaking about anti-Hebbian plasticity protects against memory interference. Just give him a minute to set up.
I'm set up. Oh, perfect. So first of all, I want to thank the organizers for the opportunity to present my research here. So Hebbian learning in general describes the synaptic strengthening between correlated neurons and is thought to be the computational backbone of memory storage and pattern completion. Hebbian learning has also been observed in inhibitory neurons, where it is thought to describe the formation of EI balance and the competition between correlated activity patterns. Anti-Hebbian E to I plasticity, on the other hand, has been observed in hippocampus memory networks, and it is still very much unclear how it contributes to network computations. Here, our hypothesis is that anti-Hebbian learning mediates the competition between anti-correlated memories, whereby excitatory neurons suppress anti-correlated neurons through an anti-Hebbian disynaptic inhibitory motif here. So to describe this principle, we took a bottom-up approach and built a recurrent EI memory network with a supralinear nonlinearity both for excitatory and inhibitory neurons, which is motivated from experimental and previous theoretical work. To store memories, we assume a synapse-type specific competitive Hebbian rule, where synapses change according to a really simple classic Hebbian rule with a BCM-like plasticity threshold, while each synapse is normalized such that the total synaptic postsynaptic weight remains constant, which we motivate by the availability of only a limited amount of synaptic resources that different synapses have to compete for. Importantly, we assume that this normalization happens for all synaptic types, therefore in a synapse-type specific way, which in our EI networks leaves us with essentially these 4 free parameters, which are the total synaptic weights for each of the synapse types. We assume that during memory storage, the network is dominated by external input, such that neural activities R become equal to the external to-be-stored patterns P, and under this condition, we can solve for the weight dynamics analytically and find that the weights become proportional to pattern covariances C. So eventually, in the steady state, the recurrent weight matrix can be written in this convenient Kronecker product form, where essentially this is really just a convenient way to write this block matrix structure here on the right. However, crucially, this form allows us to separate the computational properties of the network stored in these recurrent total synaptic weights, W, and the memory content of the network stored in the pattern covariance matrix, C. Now, one crucial limitation of this rule is that when pre- and postsynaptic neurons here are negatively correlated, their synaptic weight decays to 0 due to this plasticity BCM threshold. Now, To solve this issue, we resort to anti-Hebbian learning to exactly store these negative covariances. So at these E to I synapses, we have calcium-permeable AMPA receptors that allow for calcium influx following presynaptic glutamate release without postsynaptic depolarization, which results in long-term potentiation. However, when the postsynapse is in fact depolarized, calcium influx through these CP AMPAs is blocked, which prevents plasticity. So mathematically, we can describe this rule by simply adding a minus sign in front of the previous simple Hebbian rule, which now allows us to capture negative covariances via this anti-Hebbian rule. And let me briefly note that this is different from previous memory networks that used inhibition mostly as a globalizing, stabilizing mechanism. We here, on the other hand, do store actual memories at each E to I synapse. So together, this leaves us with a recurrent weight matrix that is composed out of two terms. The first one is a Hebbian term, all-to-all connected, which is responsible to store positive parent covariances and leads to EI balance in the network, and a second term which stores negative covariances only at excitatory onto inhibitory neurons. And note that this combination of Hebbian and anti-Hebbian learning gives us exactly this disynaptic inhibitory motive to store the negative correlations between neurons. Now, to test recall in this network, we initialized it with a noisy memory queue here in black and visualized the network's activity by projecting it onto the two first principal components of the stored patterns. So when we start the dynamics, what we observe is that a network states nicely converge to the stored memory pattern. And we further quantified this by measuring the overlap between the network states and the queued memory pattern in orange here, and all the other memory patterns in gray. And as you can see, we nicely converge to the stored pattern. So here I rearranged the otherwise random binary pixel patterns into a visually slightly easier to digest pattern. So we are already very happy. So what I'm showing you here, this is not a cherry-picked example. This is actually super robust. So when we store many different random memory queues, they all nicely converge to the queued pattern. So we're already super happy with that, having an EI, essentially Hopfield network. However, we wanted to go one step further and understand how anti-Hebbian learning actually plays into the functioning of this network. And what we did is we simulate the blockage of anti-Hebbian learning by removing all negative covariances from the learned recurrent weight matrix, here the negative, the W minus part. And as a toy example, we then stored 3 different memory patterns that all overlapped in this shared subspace, while activities in the pattern unique subspace in the bottom here were allowed to differ. So intuitively, neurons that code in the shared subspace are highly correlated and form recurrent Hebbian connections, whereas neurons that are encoded that encode the different patterns in the pattern unique subspace are highly anti-correlated, forming these anti-Hebbian net inhibitory motives. And when we block anti-Hebbian learning, this inhibition is of course lifted, which should impair recall performance. And this is exactly what we find. So when we queue each of the 3 memory patterns here with a number of memory queues, what we observe is that instead of the network recalling the queued pattern, for all the 3 queued memories, the network actually converges to a spurious attractor that is a mixture of the 3 memory patterns, which suggests that the removal of these anti-Hebbian weights impairs the recall due to a lack of competition between inhibition between these overlapping memories. Now we more systematically varied different parameters in the network, namely the overlap between stored patterns, the number of overlapping patterns, the number of stored patterns in total, and the noise level of the recall queue. And we in each time found the same general shape where the blocking of anti-Hebbian learning selectively decreased the recall performance when memory demands are high. However, when the memory demand is low, indicated by these gray areas here, network performance was very similar in control networks compared to CP-AMPA suppressed ones. So we next wanted to test this prediction in vivo experiments, and for that we replaced CLUA2 lacking AMPA receptors by CLUA2 containing receptors, which blocks the calcium influx that I mentioned before. So we therefore overexpressed the CLUA2 AMPA subunit specifically in hippocampal parvalbumin positive inhibitory neurons to block anti-Hebbian learning. which we then confirmed in slice experiments that showed that actually we see a strong decrease in anti-Hebbian long-term potentiation following this perturbation. So to test how this decrease in anti-Hebbian plasticity actually affects behavior, we next moved to two very well-established hippocampal-dependent spatial memory paradigms. And in the first task, an animal had to recall which arm of the T-maze was blocked in a previous sampling phase to receive a food reward. To manipulate the memory demand of the animal, we changed the delay period here between either 5 seconds or 30 seconds. And what we observed is that when the delay is short, in the perturbed mice they performed really comparable to control mice. However, when the memory demand is very high with this 30 seconds delay, performance was vastly decreased. And this exactly matches what we found in the model, where over time, where we interpret, we assume that the recall queue deteriorates over time, such that the shorter delays here correspond to a lower Q noise, and the longer delays correspond to a higher Q noise. And as before, performance is perturbed as performance in perturbed mice only decrease under high memory demand, that is for high memory noise levels, Q noise levels, sorry. So in the second task, which is the Morris water maze, the mouse had to find the location of a submerged escape platform, and we quantified the recall performance by measuring the time that the animal spends in the target quadrant of the maze. And what we find is that there's no difference between the CP-AMPA perturbed mice and the healthy mice or the control mice, again matching this idea that under low memory demands, there's no significant difference. However, when we relocated the escape platform to introduce a second highly overlapping competing memory, animals tend to spend less time in the new target quadrant compared to the control animals, and instead spend significantly more time in the previous target quadrant, indicating problems with recall of highly similar memories. Now, again, we compared this to our modeling work. This is exactly what we see there as well, where recall performance is high when memory demand is low, corresponding to the initial acquisition of the platform location, whereas recall performance selectively decreases in perturbed mice when memories overlap or are highly similar, which again confirms the selective recall impairment under high memory demands when blocking anti-Hebbian plasticity. So with that, let me summarize. I showed you how to construct biologically grounded EI memory networks and demonstrated that we can robustly recall different memory patterns from noisy cues. When we suppressed anti-Hebbian learning, recall performances consistently drop when memory demand is high, but remains comparable to controls when memory demand is low. And finally, we tested these predictions in two hippocampal-dependent spatial memory tasks and found very similar effects where suppression of anti-Hebbian learning impairs recall of highly similar memories. And with that, let me thank my collaborators. The whole modeling and theory part was done with Yashar Ahmadian and Matej Lengyel in Cambridge. And here, Matthew Cooper led the experimental work in the lab of David Bannerman and Edward Munn. And thank you all for your interests.
Great talk. We have time for about 2 questions or 3 questions. So I can start us with, well, I'm trying to see if anyone's coming to the mic or just moving.
I can add something.
So, you're, okay, there. Yeah, the question.
Yeah, there was a lot of people moving, so I didn't know if they were questions. So it's nice that you have very nice work. It's very interesting. I was a little bit curious on how you see the application of your model in this in vivo behavioral tasks. So I see how highly demanding tasks might fit your model. I am not sure if what you are seeing with the Morris water maze is the same type of phenomena, but that actually is less flexible, so that the animals spend more time in the previous quadrant before the reversal. It means that they actually learned that it was there but they are less flexible to try to find another solution.
Yeah.
So maybe the modeling can be applied for highly demanding memory tasks, but also it makes memories less flexible.
Yeah, absolutely. I mean, the question is how would you model this flexibility in the circuit, right? So in some sense, we are still a bit detached from the actual biology because our rate model is reasonably abstract. So in that sense, we try to really keep it rather general in terms of scanning these different aspects of memory performance by changing the overlap of the parents, the number of the parents, etc. So of course, this also plays into the flexibility of the animal to keep the old pattern and learn a new pattern. So yeah, in that sense, we try to keep it general.
Question
Okay.
in the back there.
Okay, well, thank you. Great talk. I think there are multiple brain areas that are involved in this kind of spatial task. There can be hippocampus, there can be like prefrontal cortex, there can be like retrospinal cortex.
Yeah.
I'm wondering whether you're modeling a specific brain area or you're just in general interested in the interaction between excitatory and inhibitory.
So this perturbation in the behavioral data, this was specifically for parvalbumin cells in area CA3 of the hippocampus. So in that sense, you really target that area, and that's also where we think this highly, strongly recurrent networks live that enable the memory recall.
Thank you.
More of a theoretical question.
Yeah.
So this is essentially, as I understand, a Hopfield model with Hebbian learning. And what is nice with Hopfield model is that we can quantify mathematically the memory capabilities of the model.
Yeah.
Do you know if adding Hebbian learning increases those capabilities, not only for recall, but of the amount of storage in the network?
If Hebbian capabilities. So I mean, well, technically, also Hopfield networks, in a sense, use Hebbian learning, right? We use the same outer product between the storage patterns, right?
If you add anti-Hebbian, do you increase the storage capabilities?
Well, yeah. So essentially, in the Hopfield network, we automatically also use anti-Hebbian learning because we do not have this Dalian constraint in our recurrent weights, which, however, we actually enforce here. So in that sense, we have some more theoretical insight into this, but essentially this is a bit of a convoluted way to approximate a Hopfield network while maintaining key biological principles, that is EI balance, these superlinear activation functions, Dalian synaptic weights. Yeah, so in that sense, with some caveats, think of this as an EI approximation of a Hopfield network.
Okay.
Yeah.
Thank you very much.
Thank you.
Let's thank the speaker again. We should move on to the next talk. Our next speaker is Mark Plitt. He's a postdoctoral researcher at UC Berkeley, and he'll be speaking about neuromodulatory control of rapid plasticity in a head direction circuit. Also, I have someone's wallet who left it here. If you are Manuel Baran, I have your wallet, so come up and get it when you are ready.
All right, thanks everyone. Hopefully I'll touch on some related themes in a different context, so like parallel fiber systems and actually some anti-Hebbian learning. And thanks to the organizers and reviewers for giving me this opportunity. I also want to start off by acknowledging the folks who contributed to this work, both former and current members of the Fisher Lab, as well as our friends and collaborators, Dan Turner-Evans and Vivek Jayaraman. Today I'm going to tell you how the fly navigation circuitry uses the neuromodulator octopamine to drive synaptic coincidence detection. So navigating animals need to maintain an internal representation of their heading in the environment. And in every navigating species that people have studied so far, the basic neural algorithm by which animals do this is the same. And that's with a population of head direction neurons. Each head direction neuron fires most when when the animal is facing a particular orientation relative to salient landmarks like the sun. Different cells have distinct preferred orientations. And as a population, these cells tile the different orientations to form a landmark-based neural compass. And for this compass to work well, these head direction circuits need to combine information about self-motion with sensory information about reliable landmarks. And despite the decades of foundational work in these circuits in numerous species, we still don't have an answer to this fundamental question, which is how do head direction circuits learn about landmarks? So we took advantage of the compact head direction circuitry in the fly to start to understand this learning process at a mechanistic level. The key region of the head direction circuit in flies is this ring-shaped structure in the middle of the brain called the ellipsoid body. And the main head direction neurons are called EPGs. Each EPG has a dendritic arbor that's confined to a single wedge of the ellipsoid body, and as a population, they tile the different wedges with their dendrites. And these neurons have striking dynamics. If you record from them while the fly is behaving, what you see is a localized bump of increased neural activity in these dendrites that swings around the network like the needle of a compass to track the fly's orientation. And in this example, the angular offset between the landmark around the fly and the angle of the bump in the brain is 0 radians. But if we record from other flies, we see that they actually have random angular offsets, but they still accurately integrate heading, and these offsets are set by the plasticity between visual neurons and head direction neurons. And these plastic landmark inputs provide a critical error-correcting signal. For example, if flies walk in the darkness, we and others have observed that there's still a bump of increased neural activity, and it roughly tracks the fly's movements, but it quickly accumulates error. And so when the landmark becomes available again, the bump is pulled back to its previous offset, and the circuit starts accurately integrating once more. So this anchoring visual information comes into the ellipsoid body through a set of inhibitory neurons called ring neurons. Here I'm showing an example of one type of visual ring neuron, and then all of the neurons of that subtype. Each one of these neurons has a ring-shaped axon that contacts every EPG, forming all-to-all connectivity from the EPGs from the ring neurons to the EPGs. And from previous work, we know that this class of ring neurons is essentially a set of vertical bar detectors with receptive fields that tile visual space, which means that each EPG receives inhibitory synaptic input about every orientation. So let's imagine that light from the landmark hits the retina in the receptive field of this ring neuron, causing it to fire. It's believed that the synaptic weights from this ring neuron onto the postsynaptic EPGs are determined by associative plasticity. So if these EPGs highlighted in red happen to also be active the first time the fly sees the stimulus, then the connections between these EPGs and this ring neuron will undergo depression. And now the next time that the stimulus is in the receptive field of this ring neuron, only this wedge of the EPGs will be disinhibited, and all competing locations around the ring will be suppressed. So now when the fly turns, the landmark will move into the receptive field of other ring neurons, and self-motion will cause the EPG bump to slide over. And the same learning will then cause these synapses to depress. And if you repeat this at all orientations, now you have the perfect synaptic structure for accurately integrating heading. And this plasticity rule is computationally straightforward. If two neurons are coactive, the synapses between them depress. But we actually have no idea how this works, and it's my strong belief that by understanding the brain's specific solution to this under-constrained problem, we can learn a lot about the computational and physiological principles here. So as a first step, we want to understand how coincidence detection is performed. How do the ring and EPG neurons detect that they were both active? In other systems, this type of inhibitory plasticity is often accomplished by the active postsynaptic neuron sending a retrograde signal back to the presynaptic neuron, which will undergo depression if it is also active. However, EPGs likely don't produce any known retrograde signal. So inspired by work in other invertebrates, we thought that maybe a neuromodulatory neuron could function like a retrograde signal. And it turns out there's a relatively unstudied octopaminergic neuron that is a perfect candidate. That neuron is called EL. ELs have wedge-shaped arbors that tile the ellipsoid body, which is incredibly similar to the anatomy I just showed you for EPGs. And from connectomic data, we know that one of the main inputs to EL are excitatory synapses from EPGs, and their main outputs are sensory ring neuron axons in the same wedge, which suggests that EL may convey a copy of the EPG head direction bump back onto ring neuron axons in the form of octopamine. So we just tested if that was the case. We had flies walk on an air-supported ball in virtual reality under a 2-photon microscope. Meanwhile, we simultaneously imaged the activity of EPGs and ELs using 2 different colored calcium indicators. First, let's just consider the EPG data. Imagine that we're looking down at the ellipsoid body in our imaging field of view, and that I cut open the ring along this 0 to 2 pi line and splay the network out to form a column vector. Now we can plot the whole network's activity over time as a heat map. So hopefully what you can see is that there's a clear bump of increased neural activity that drifts around the network. And if I superimpose the location of the cue, you can see that these two things move in lockstep with one another. On the right, I have a histogram of the offset values between the orientation of the cue and the location of the bump around the ring. You can see that we have a nice peaked distribution indicating a consistent head direction code. Now, if we look at the EL neurons using the same analysis, we see that this is essentially a copy of the head direction signal. Both populations show a clear bump of activity that tracks the cue at the same bump cue offset. And when we measure the angular distance between these two bumps, we see that regardless of whether the fly is walking in darkness or in closed loop, these two bumps are very close to each other. So EL really is a near copy of EPG activity. And to close the loop, we confirmed using an octopamine sensor, GrabOA, that the ring neuron axons receive a compartmentalized bump of octopamine. And sequencing data shows that ring neurons express multiple octopamine receptors, meaning they're competent to respond to this localized signal. So we have this 3-neuron loop where we hypothesized that synaptic depression occurs if all 3 neurons are coactive. And in data I don't have time to show you, we demonstrated that EL octopamine synthesis is necessary for accurate head direction tracking by EPGs, which suggests that this neuromodulatory loop must be intact for plasticity to occur. But really, if this retrograde-like signal from EL is the true instructive plasticity cue, then the hypothesis is actually that only the ring neuron and the neuromodulatory neuron are directly involved in plasticity. And so to test that idea, we need a way to activate these 2 neurons while ensuring that the EPGs are silent. So we did that using a combination of visual cues and VR to drive ring neurons, and then multiple optogenetic ion channels. We used an excitatory opsin to drive EL neurons, and an inhibitory opsin to silence EPGs, and at the same time we'll also image EPGs with GCamp. So we can use one laser for imaging, which importantly doesn't strongly activate either optogenetic channel, and then we use a different independently controllable laser that can activate both of our optogenetic channels at specific times and locations. So let's walk through one of these experiments. Before we do any optogenetics, we simply let the fly walk in VR, and we'll measure the spontaneous bump cue offset that the network learns. So this is an example of one of those sessions. You can see a clear bump of EPG activity that tracks the bar at an offset near negative pi/2. And we can succinctly represent this histogram by its resultant vector, which points in the direction of the average offset, and its length tells us how consistent that representation was. So next we'll use our optogenetic laser to try to entrain a new bump cue offset, in this case at 0 radians. We do that by showing the stimulus at a particular orientation, and then using our optogenetic laser to stimulate only the wedge where we want the bump to be. And again, this laser will activate EL neurons while silencing EPG neurons only in that location. And so we need to teach the network a consistent mapping, so we repeat this at multiple orientations. So we start off with the bar here and the bump here, and then we move the bar around the fly, and we shift our optogenetic laser so that we maintain the same offset. And then we do that again, and we keep doing it until we go all the way around the network and repeat that 5 times. And then we'll image and see if the network retains the offset that we entrained. So now this is data from the same fly just a few moments after I did that pairing, and you can see that the offset has shifted, so it's almost exactly at 0. The bump and the cue are exactly on top of each other, and it's accurately tracking heading. This resultant vector points almost exactly at 0. So now in the same fly, we'll see if we can do it again and shift the offset this time to pi radians and then record. And again, now in the same fly, just a few moments later, we've shifted the offset completely across the network. So this effect was incredibly consistent across flies. So here's that polar summary from the example fly that I showed you, and then just the plasticity sessions from all of the flies. So we can reliably drive plasticity in this circuit just by activating the ring and EL neurons. But is this activation of EL also necessary for plasticity? So previous work that really inspired many of our experiments showed that you can drive plasticity in this circuit through pairing only the EPG and ring neuron activity. We replicated that finding using our variant of this protocol, which you can see by this nice separation of the red and blue lines. But our hypothesis is that this protocol works because it recruits this local EL feedback. So if we break that feedback loop by silencing the EL neurons, we now completely block plasticity, which you can see by the interspersed red and blue lines, and flies still learn the original bump cue offset, but it doesn't change after our pairing. So where does that leave us? We started off with a hypothesized plasticity rule, where synapses between coactive ring and EPG neurons depress in order to tether the head direction code to landmarks. And then I showed you that coincidence detection works through a surprising and completely novel mechanism where postsynaptic activity is relayed through a neuromodulatory neuron back onto the presynaptic terminals, a plasticity motif that might be hiding in other circuits as well. So this is the deepest mechanistic description of head direction plasticity in any organism that we know of so far. And it's also an entirely new mechanism for doing associative plasticity at inhibitory synapses, a topic that's not really that well understood. And now that we have a handle on how to reliably drive plasticity in the circuit, we can begin to study how the biochemistry of the ring neurons actually calculates this correlation between their own activity and the octopamine. And we're fascinated to try to understand the adaptations that are required for doing rapid scene learning using a neuromodulator, which people traditionally think of as a slow signal. And perhaps most importantly, can we find the principles that are shared between this mechanism and mechanisms used in, say, reward learning circuits where the roles of monoamines are much better established? And with that, I'll wrap up and make sure to thank everyone involved in this project. And I'll happy to take any questions from time to minutes.
We have time for 2 or 3 questions. I'll ask a quick one to get started. You kind of alluded to it, but neurotransmitters are typically thought to operate very slowly for this kind of rapid learning. Do you have directions that you're moving in to try to understand how this can still work even with this relatively slow signal?
Yeah, so I mentioned that there's a handful of octopine receptors that are expressed in the ring neurons, and actually 2 of them have opposing effects on cyclic AMP. So one of them is a GS-coupled receptor, and the other most strongly expressed one is a GI-coupled receptor. And Gabby Mayman has also done some of this work, but we imaged cyclic AMP in the ring neurons, and there's a bump of increased cyclic AMP that tracks the fly's movement, so at least the second messenger can sort of keep up with the behavior. So they might be sort of specific ways they tune the relative expression of these receptors to make that possible. Yeah.
Question here.
That was a great talk. I'm just wondering, is there some extra value of adding this extra EL neuron in between the 2 as opposed to just a direct feedback from EPG neurons to the ring neurons? Like, for example, is it adding some information? Is there some added, regulatory input to that cell? Is there anything that, is computationally valuable by adding that extra layer?
Yeah, so there's a couple of things. I think the most compelling idea is that it allows you to regulate plasticity without affecting maybe the fine-tuned architecture that's needed here for smooth head direction tracking. And sort of in line with that idea, the EL neurons get different inputs than the EPGs that are involved in circadian rhythms and sleep, and they also express distinct GPCRs from the EPGs, so they can respond differently to cues like serotonin. Yeah, and yeah, there's other ideas too, but I think that one's the most compelling. Yeah.
I'm going to stop the questions here. There are a few others, but hopefully you'll find each other just because we're running a little over. So let's thank our speaker again. This will be our last talk of the session. It's from Adrian Holtrup. He is a student working at Harvard Medical School and also the University of Munster, and he'll be speaking about some parallel fiber subsampling in the cerebellar cortex as a substrate for diverse representational learning.
Thank you. Good afternoon. Thanks to the organizers for the opportunity to speak here. My name is Adrian Holtrup. I will present on a study that I worked on in the lab of Wei-Cheng Li as part of my medical thesis. Special thanks to Ramin Khadje for his substantial help with this project, as well as to Ashok Ladwin Kumar, Nate Sartle, and further members of the Li and Regia labs. Also, I'm grateful for generous funding from Boehringer Ingelheim Fonds. I'll start with an analogy to geography that may help grasp the fascination in connectomics. If we take a look at this world map from 1800, we see the outlines are roughly correct, but the map wouldn't have been of much help to get from the hotel to the conference venue this morning. 2 centuries later, we have developed satellite service like Google Earth, where with just a few scrolls, we can go from planet Earth to exactly the place we're just in. A similar thing happened in neuroscience. This is a drawing by Ramon y Cajal from 1888, one of the first detailed depictions of cerebellar anatomy. And nowadays, thanks to advances in large-scale connectomics, we have the fascinating opportunity to look at this circuit at synapse resolution. And my talk will focus on how this changes our understanding of this circuit. So the microcircuit of the cerebellum comprises 2 very distinct inputs. First input is conveyed through mossy fibers that synapse onto a vast number of granule cells. These granule cells extend parallel fibers in the molecular layer, hundreds of thousands of which contact the dendritic trees of Purkinje cells, the sole output neuron of the cerebellar cortex. Second, each Purkinje cell is directly innervated by a single climbing fiber, a highly potent input that instructs plasticity at the parallel fiber to Purkinje cell synapses. This formation has been central to the foundational models that demonstrated how, through learning, the climbing fiber shapes the response of the Purkinje cell to specific patterns of inputs. And these canonical models have earned the cerebellum a place among the most attractive circuits to study the relation between circuitry and function. But while successful, they did not take into account several key features, 3 of which I will focus on in the following. First is the idea of microzones. Purkinje cells are organized in sagittal zones where they receive highly correlated and co-tuned climbing fiber inputs. This is evidenced by a large body of both functional and anatomical studies, including, for example, this recent work that shows how calcium signals in Purkinje cell dendrites evoked by climbing fiber inputs are correlated within sagittal zones. Further, again within a sagittal zone, Purkinje cells converge at the deep cerebellar nuclei, the output stage of the cerebellum. These observations suggest that the functional unit of the cerebellar cortex, instead of individual cells, is rather a population of Purkinje cells within a sagittal zone. The second is inhibitory interneurons. The molecular layer comprises a dense network of GABAergic molecular layer interneurons, or MLIs for short. Recently, they've been shown to group into 2 subtypes, MLI1s that inhibit Purkinje cells, and MLI2s that inhibit MLI1s, thereby disinhibiting Purkinje cells. Notably, like Purkinje cells, they receive dense parallel fiber inputs with synapses that are subject to plasticity, thus potentially playing a critical role in cerebellar learning. The third feature is what I refer to as partial connectivity. To illustrate this, I'm going to switch from this schematic to one that views the circuit in the sagittal plane. Here, the triangles denote parallel fiber synapses. Now, interestingly, prior evidence suggests that the connectivity here is in fact partial, meaning that a considerable number of parallel fibers will pass through the dendritic tree of the postsynaptic neuron without making a synapse. This is a key anatomical feature that is left out in classical models. But now that we have a group of Purkinje cells and MLIs potentially forming a functional unit, partial connectivity becomes an interesting observation, as it suggests there may be differential innervation through parallel fibers in the molecular layer, possibly reflecting an organizational principle of cerebellar learning. But the rules governing parallel fiber innervation are unclear. So to address this question, we made use of a volumetric EM dataset at Synapse resolution that features reconstructed Purkinje cells, MLIs, and parallel fibers, a few example cells illustrated here. A natural low-order approach when analyzing connectivity is to first search for structure at the level of cell types. Therefore, we first asked if parallel fiber inputs to MLI1s are different from those to MLI2s. Here I show a synaptic connectivity matrix where each column belongs to a parallel fiber and the rows correspond to MLIs. We sorted the parallel fibers by their affinity to MLI2s. If now we insert the MLI1s into this matrix, we observe this striking dichotomy, meaning that we can hardly find a parallel fiber that synapses onto both MLI1s and MLI2s. So the MLI subtypes here receive almost mutually exclusive inputs, which to us was a proof of principle that indeed innervation through parallel fibers is organized. We then wanted to see if there's also a relation in innervation between MLIs and Purkinje cells. Again, we look at the connectivity matrix, and this time we sort the parallel fibers by their affinity to Purkinje cells. If now we add the MLI1s, we see a trend that those parallel fibers undersampled by Purkinje cells are more densely sampled by MLI1s. For MLI2s, the situation is the opposite, meaning that MLI2s connect mostly to parallel fibers oversampled by Purkinje cells. Now, often in connectomics, structure arises from spatial biases. For example, for this Purkinje cell, there are some parallel fibers here aligned with the viewing axis that don't even cross its dendritic tree and will certainly not make a synapse. So to better disentangle if the patterns that we observe are due to the spatial distribution of the cells or due to wiring cell activity, for each cell we computed which parallel fibers get close to its dendrites below a given threshold. We call this touches. Conditioning the analysis on touches will then allow us to eliminate spatial biases. If now we consider a second cell for comparison, say this MLI1, we're interested in those parallel fibers that touch both cells, the shared touches. Within the set of shared touches, we then investigate shared synapses, deriving a metric that accounts for the input overlap of these two cells unbiased by their spatial distribution. Now remember the dichotomous innervation of the MLI subtypes. We want to apply the touch analysis to this result. First, for different combinations of pairs of MLIs, we compare the number of shared synapses. As would be expected from the connectivity matrix, this value is low for MLI1, MLI2 pairs compared to pairs of the same subtype. However, this does not hold for the shared touches. Now, this was an intriguing finding because it implies that even though MLI1s and MLI2s have access to the same set of parallel fibers, they selectively sample different inputs. If we then divide the shared synapses by shared touches for each pair, we obtain a compact, spatially unbiased metric that we call the relative overlap. Likewise, we applied the touch analysis to our observations regarding Purkinje cell innervation. Here, for individual parallel fibers, we correlate the synapses per touches to Purkinje cells to the synapses per touches to MLIs. For MLI1s, we observe a negative correlation, for MLI2s a positive one, confirming that our previous observations are explained by wiring selectivity rather than the spatial distribution of the cells. So the touch analysis points to specific rules that govern parallel fiber innervation. Nevertheless, so far, we have looked at correlations in innervation between cell types, but this does not yet inform us about the origin of these correlations. A natural but not yet implied assumption is that all of these three cell types sample parallel fibers according to some cell type-specific rule. If so, we would expect that two representatives of the same cell type have a higher relative overlap than predicted by chance. We investigated this by comparing the observed connectivity to random touch-constrained Bernoulli models and found that for MLI1s as well as MLI2s, this is indeed the case. But pairs of Purkinje cells had just the same relative overlap as the shuffle. Another approach that works without a shuffle is to cross-validate the connectivity. We divide cells of a given type into two partitions and see if we can infer parallel fiber innervation of the second partition from that of the first partition. Again, for MLIs, this is the case, but not for Purkinje cells. Across a series of other modalities of analysis, examples include the variance explained by principal components or pairwise mutual information. The observation holds that parallel fiber Purkinje cell connectivity is well explained by the random Bernoulli model. So let's collect the results so far. First, we observed that inputs to subtypes of MLIs are mutually exclusive. A similar relation, but rather gradual, held for MLI1s and Purkinje cells. MLI2s, on the contrary, tended to share inputs with Purkinje cells. We then investigated intra-cell type connectivity and saw unmistakable signs of selectivity in inputs to MLI1s and to MLI2s, but the wiring diagram between parallel fibers and Purkinje cells seemed unstructured. Now you may be wondering how unstructured parallel fiber Purkinje cell connectivity can be in line with the correlations that we've seen earlier. And there are different possible explanations. I'm happy to discuss them after, but the hypothesis which to us seems most reasonable is that parallel fiber Purkinje cell connectivity is indeed unstructured and results in a local connectivity distribution where, just by chance, some parallel fibers are over, others undersampled. MLIs then sample their parallel fibers depending on this distribution. Now, critically, such a model is at odds with hypotheses that interpret partial connectivity as a result of learning and instead favors one where partial connectivity is a substrate of learning. So this made us curious if unstructured parallel fiber Purkinje cell connectivity could have an advantage for cerebellar learning. So here I will leave the connectivity analysis, and for the last bit of my talk, we'll focus on this particular question. In a model with a single Purkinje cell, it's trivial that arbitrary omission of parallel fibers will simply lose useful information. But if we consider an ensemble of Purkinje cells, the outputs of which are pooled, partial connectivity may diversify otherwise redundant Purkinje cells. To investigate this systematically, we introduce a parameter rho that determines the connection probability between each parallel fiber and Purkinje cell. Now, how can we train such an ensemble? The challenge that comes with multiple Purkinje cells is that the feedback signal, the climbing fiber signal, will be generated based on the ensemble output and hence be the same for all Purkinje cells. But for an ensemble mechanism to work, it's critical that the ensemble members are trained individually. Therefore, we reasoned that particularly active Purkinje cells, as denoted by color here, should be punished more strongly for their larger contribution to the ensemble error. To this end, in line with studies in cerebellar slices, we added a third factor to the canonical two-factor learning rule that accounts for the postsynaptic activity. With this adapted learning rule, we investigated the performance and classification tasks. When considering a single Purkinje cell, the error rate trivially decreases with increasing connection probability rho. But for ensembles, there is indeed an optimum at partial connectivity trading off input dimensionality against ensemble diversity. Now, critically, this effect depends on the modification of the learning rule. If instead we use a simple two-factor learning rule, the ensemble advantage is blighted. So summing together, this demonstrates that, in fact, partial connectivity may help the cerebellum to diversify the network, similar to what is used as ensemble techniques in machine learning. Regarding ensembling in the cerebellum, there is independent but related work by Ben Rubin and Cengiz Palavan, and I encourage you to check out their poster tomorrow. That said, thank you, and I'm happy to take questions.
I know that question session is going to hold everyone up from lunch, so please try to, if you're going to exit, please do so quietly, just so we can maintain the integrity of the question session, and we have time for 1 or 2 questions.
Very fascinating results. If I understand correctly, you've got 2 classes of parallel fibers and therefore 2 classes of granule cell that synapse onto the different types of interneuron. So if I understood right, you're saying they're not molecularly different types of granule cell, but they are somehow informed about this by the fact that if you synapse more onto Purkinje cells, you'll become a type 1 granule cell. If you synapse less, you'll become a type 2 granule cell. So how does that work? Is there a signal that goes back to the soma along the axon?
So currently I wouldn't really call them types because we don't know about any molecular characteristics or anything. It's just that we observe that those parallel fibers or granule cells connecting to MLI1s usually don't connect to MLI2s and the other way around. Concerning your question, we have checked if these populations have different mossy fiber inputs, and as far as we could tell from what we've seen in our connectome, this is not the case. But we cannot say anything about any transcriptomic or genetic profile of these cell populations. An idea that could explain how this connectivity comes about is that there is some sort of local signaling in the axons, in the parallel fibers, that signals that if there is a parallel fiber MLI2 synapse, there will not be a parallel fiber MLI1 synapse in the proximity, but that's just speculation at this point.
Thank you.
So we can take one more question, and then we'll break for lunch.
Great talk. So you talked about microzones, but you didn't come back to that, the microzones. So what I want to know is that the connectivity of parallel fibers to MLI1, which you said is not overlapping with connectivity to MLI2. Does that happen in different microzones?
No, yeah, that's an excellent question. So we don't have, in our data set, we cannot with precision determine where a microzone would have a border, but the data set is likely within a microzone because it has an extent of about 40 microns in the z-axis, and a microzone is usually considered to be around 100 to 200 microns. So it's probably within a microzone, but we cannot say anything about the extent or the connectivity across several microzones.
Thank you.
Okay, let's thank the speaker. So there will be a lunch break. Poster session starts at 1:15, and there are sandwiches downstairs on the first floor. I'll do it. Oh, yeah, no, I said his name. I don't think he's in the room anymore, though. Oh, yeah. And any friend of Manuel Baran, please come grab his wallet for him. Or if you are him. Huh? Oh, there's your wallet. Yeah, there you go. Yeah, no problem. Yeah, no problem.